right. So thank you, James, for organizing this symposium today. I'm going to talk about a new dimensional multi-site neuroimaging reference system on which we are working at the moment at the Center for Biomedical Image Computing and Analytics at the University of Pennsylvania. This new reference system is going to enable us to better understand the heterogeneity of brain aging. So I hope that I know now. Okay. All right. It's well known that age is associated with a lot of diseases that are heterogeneous in nature, such as neurodegenerative disorders. And we know that other factors like genetics or lifestyle risk factors would modify the brain aging trajectory of an individual. So there is a big evidence that people are not aging in the same way, but rather in a heterogeneous, we're taking different trajectories. These days we are using big data, we are using neuroimaging large-scale data with complex multiparametric signals, which we want to capture in a way that we keep the information and we go out of the curves of the dimensionality. So our approach is to use machine learning method to reduce the dimensions and come up with imaging markers that are concise yet informative. So in this conference system, we are using the machine learning, large-scale studies, and neuroimaging. Okay. The basic idea of the eye staging reference system is to come up with different dimensions that would tell us how the brain is changing through either disease or age. For example, we are using what we call the SPARE indices, in which we are capturing change related to disease like Alzheimer's disease with a SPARE AD index. We have developed also an index which we call the SPARE BA index, which is capturing age-related atrophy. The same technique, which is relying on classification, can be applied for other neuropsychiatric disorders, so to detect change related to schizophrenia, for example. There is also a SPARE schizophrenia index, which we have developed. Other dimensions that are represented in the eye staging reference system are related to vascular brain injury. We are using also machine learning methods to detect white matter hyperintensities. We are downsampling the neuroimaging data using data-driven approaches to come up with functional networks using sparse connectivity patterns, for example, or non-negative matrix factorization to represent structural covariance patterns. We are also using clustering algorithms to understand subtypes of disease like Alzheimer's disease or subtypes of deviation from typical brain aging, what we call advanced brain aging. I will be giving examples about all these dimensions in my talk today. So towards this eye staging reference system, we are building the eye staging consortium. The eye staging consortium is consisting of a lot of studies from all around the world with studies covering the heterogeneous aspect of brain aging from the PLSA study, for example, which is following healthy individuals in the United States of America. Studies like ADNI or PEN-ADC are focusing on Alzheimer's disease. We have samples from the general population like the SHIP study. So we are pooling all these data together, and all of the participants of the consortium, at the moment there are 11,000 participants having structural MRI data with about 15,000 scans, so we have some longitudinal subsets. We have also resting state of MRI data for a couple of studies in addition to amyloid PET scans. So I will be now giving a couple of examples of the dimensions which we are representing in the eye staging reference system. Since it is an aging consortium, the first dimension which we are interested in is, of course, the brain aging dimension. And this is an example here which is showing the SPARE BA index, an imaging marker to detect age-related atrophy in the SHIP study with 2,700 individuals. We show here that the brain age atrophy is is getting worse over time. And brain aging is starting to happen by the year of 30. Before that, it's more stable. And we have shown that a lot of risk factors are leading to more atrophy patterns than expected. What is, for example, a risk factor could be hypertension. So if you have hypertension, you might get an older brain than it should be. Other dimension I'm going to show today is the AD dimension. We are capturing the AD-like patterns of atrophy with the SPARE AD index. It's relying on a similar technique classification model trained by the ADNI cohort, and the SPARE AD index is capturing atrophy in AD-related regions and is detecting conversion from healthy control status towards mild cognitive impairment and from mild cognitive impairment towards Alzheimer's disease. It is an imaging marker. If we include further information like cognitive status or genetic information like ApoE, which is the genotype associated with sporadic Alzheimer's disease, this conversion detection becomes better. Okay, a third dimension I'm representing is the vascular dimension. White matter hyperintensities are showing this particular change, which is related in general to vascular disease. We have studied that extensively and shown that before the age of 40, white matter hyperintensities in the brain are rare. They become more systematic by the age of 60, and by the age of 80, the probability to have ventricular white matter hyperintensities become around 40%. Okay, so here is an example of the iStaging reference system where we pool data from 6 studies together. To the left, we show how clinical diagnosis is leaving traces in all these 3 dimensions, on the SPARE AD dimension, on the SPARE BA dimension, and on the vascular brain injury dimension. So the blue are the healthy control individuals, and from MCI to AD, we see that the 3 dimensions are already associated with the clinical diagnosis. On the right, we see how the ApoE genotype is leaving traces mainly on the AD dimension, to some extent on the BA dimension, the brain aging, but to lesser extent on the vascular brain injury dimension. So the question that can be asked now, is there an association between these dimensions? And the answer is yes. So we have shown that having more white matter hyperintensities in the brain is leading to have more aging atrophy, so they might be leading to accelerated brain aging and to have more atrophy in AD-related regions. So we have represented in this study high white matter hyperintensity load to be people having more than 80% of total white matter hyperintensity volume. And those people, according to our calculation in that study, showed that having 1 milliliter of white matter hyperintensity would mean to have around 1 year older brain. Okay, so the other question which we want to answer now, is there heterogeneity in these dimensions? And the answer is yes. So in the vascular dimension, we have applied recently data-driven approach, non-negative matrix factorization. This is a paper just got accepted in neurology. And we decomposed white matter hyperintensities maps from the SHIP study of health in Pomerania with around 1,800 individuals. What we got are 4 very interesting components. One of them was, yes, the 4 components, they are overlapping with the clinical definition of white matter hyperintensities in the deep and periventricular regions. So the deep component is here in red, and the periventricular regions were split further in 3 components that are anterior periventricular, posterior periventricular, and dorsal periventricular. Very exciting finding was here that they were associated with different risk factors that are leading to disease, like AD genetics was associated more with the dorsal component, while the anterior component was associated with blood pressure and appeared at earlier life. So this differential association with risk factors is leading us to believe that they have different etiologies, which is also confirmed in other pathological studies. And in this slide, I'm just showing quickly that these components are actually representing subtypes as well as staging. I mean, in the SHIP study, which was the discovery sample, 15% of these people were from these participants of the SHIP study were not fitting in the progression model, while 30% of the individuals were more according to anterior first and then posterior second, and so on and so forth. Taking the longitudinal sample from another study, the PLSA study, showed that the progression model was more fitting to the data. Okay, so now heterogeneity in the AD dimension. We have applied clustering algorithm on the ADNI cohort for MCI and AD cases, and what we found are 4 very interesting clusters that are fitting in the clinical subtypes of Alzheimer's disease, how they are defined at the moment. One group, for example, number 3, had relatively more executive function loss, more widespread cortical atrophy, and more white matter hyperintensities. Probably they are representing mixed pathology, which could be labeled in the clinic as Alzheimer's disease. Group number 2 was having more pronounced memory loss and less white matter hyperintensities, and they have the worst survival curves in terms of conversion. Probably they are representing the true AD pathology. We know that the AD diagnosis is very heterogeneous, clinical AD diagnosis, so that fits very much into the model. So I don't know. All right. So heterogeneity in the advanced brain aging dimension. This is a recent study which just got accepted in neurobiology of aging, where we are using 2 dimensions in the eye staging reference system. We are predicting age from structural MRI data, we are predicting age from functional MRI data, and we are defining people who are advanced brain agers as those who have more brain change related to according to their age rather than it should be. And taking the people who are advancing in 1 dimension at least all together and clustering them showed us the following. Okay, the following result. So we got 5 interesting groups where we have seen distinct pattern of different change between the individuals who are advanced in terms of brain aging. One interesting group resembled early stages of Alzheimer's disease with hippocampal atrophy and increased medial temporal connectivity. Other group showed more decreased motor connectivity. So there is very good evidence of heterogeneous brain change according to age, and people are not aging exactly in the same way. I will skip this one now. All right. So I have given examples of how we can reduce the dimensions using machine learning into different ways. One of them is using informative markers, the SPARE indices, detecting pathological change, also using machine learning segmentation in this point, or clustering. We are working now more and more to integrate different modalities together, amyloid as well as resting state of MRI data with structural MRI, to understand better the heterogeneity of brain aging. I want to thank the eye staging team who are participating, and all studies and universities who are contributing to the eye staging consortium. I want also to thank you all for your hearing, and very much happy to see your questions.
Thanks very much.